Hello and welcome back. Today we are tackling defense against infectious disease. So we are going to talk about your immune system mostly. We're going to get a lot of the basics down. In year 2, there's a lot of discussion about taking this further. There's a whole nother topic dedicated to the immune system, but this should cover all of your basics of defense. So let's get started. Let's start with just what is a pathogen? And a pathogen is any disease-causing agent. So they can be bacteria, viruses, fungi, or protozoa. Just some basic info about them, right? Bacteria are prokaryotes. This is like what causes food poisoning. Viruses are non-living. They mutate and evolve very quickly. This is like the flu, which is why you need a flu shot every year, because the virus itself evolves every year and mutates every year, which is crazy, but it does. A fungus is like athlete's foot. It reproduces with spores, which is unique to a fungus, and it is a eukaryote. And then protozoa is like a simple parasite, like malaria. Different methods of transmission, there's a number of them, but you can inhale droplets, like if someone sneezes around you and you like see the droplets in the air and then you breathe those in, which is gross, I know, but that's one method of transmission. There's also direct contact, right? If you touch an infected person where they're infected and you also have like a cut or something, it can get into your system. Bodily fluids, right? Like spit or anything like that. Animal vectors, right? If you get bit by like a rabid raccoon or something. Blood contact, if your blood contacts another person's blood, that's not good. And then ingested or swallowed. So if you eat like raw chicken, there's a chance that you could get salmonella because the bacteria gets cooked out. Of course, not all chicken has salmonella, but if it did, it would get cooked out. But if you eat it raw, there's a chance that you could get the bacteria and then it would affect you. So let's talk about first line of defense. This is like surface barriers. This is like very just like a broad sense of defense. This is like your skin and mucous membranes, right? Your skin is going to just like prevent anything from getting in. It doesn't really care if it's a virus or a bacteria or anything like that. It just exists to prevent entry, right? The cells are super close together. And the surface cells are dead, so even if something did get into a surface cell, it can't do anything because the cell is already dead. It contains keratin, which is pretty strong. It secretes oil to inhibit bacteria and lower the pH again, which would prevent bacteria from being able to replicate. And it has its own microorganisms on it, like very normal to have microorganisms on your skin, but they're going to compete with other pathogens trying to get into the skin, and so that's another way that the skin protects you. And then mucous membranes, this is like in your mouth and your nose, if you're biologically female, in your vagina as well. This releases fluids pretty consistently to wash away pathogens and also secretes enzymes like lysozyme, which causes a cell to lysis or like explode. And may be ciliated, like it has like little hairs like in your nose, which aids in removal of any pathogenic substance. The second line of defense is also non-specific, okay? So again, it's very broad. This is going to happen regardless of what type of microorganism is attacking. But this includes like fever, inflammation, phagocytic leukocytes, and antimicrobial proteins. So this is just like a very general, like something happened, we're going to try and get rid of it. I don't care what it is, but get it out of the body, right? So like if you're sick, one of the first clues of getting sick is you get a fever because your body is running hot to try and kill whatever it is that's in your body. When there is something that, like in this case, it looks like something is cutting through the skin and there's bacteria on there, there's going to be a chemical signal that's released And from the blood vessel, you're going to get phagocytes coming out and being like, no, 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 I don't want this. And they're going to like eat up all of this bacteria and get rid of it and try and remove any pathogen as quickly as possible to prevent spread. So how do these leukocytes actually ingest pathogens? There's a number of ways. There's chemical attraction. So when the damaged tissues release those chemicals, they are kind of just like magnetically pulled to them, and then we'll begin eating the pathogens. There's amoeboid action, which is they just circulate in the blood, but may move into body tissues if there's an infection. Endocytosis, where pathogens are engulfed, and then they form a vesicle around that pathogen, and so it's like preventing it from moving on to anywhere else. And then enzymatic digestion, where the vesicle will then fuse with the lysosome, and the lysosome will then digest the pathogen. This is referred to as phagocytosis, right? This is like actually what that means, is the enzymatic digestion. So antibody production, let's get into specifics on what these two terms mean, because we're going to use them a lot and I don't want you to get them confused because they do sound similar. So there's antigen and antibody. So an antigen is a substance that the body recognizes as foreign and will then evoke an immune response. Wow, okay. This is something that will then trigger a response from a lymphocyte, which is a type of white blood cell. Okay, so an antigen is foreign, an antibody is what your body produces. Okay, so a protein produced by certain white blood cells, typically B cells and plasma cells, in response to an antigen. So an antibody gets produced only when an antigen comes into the picture. The antibodies are specific to the antigen. Okay, so antibody is in your own body, and then an antigen is a foreign substance. So how do antibodies even help? Well, they make the pathogen more recognizable to the phagocytes, and then the phagocytes can come in and engulf them and get rid of them. It also prevents viruses from docking on host cells. Remember that viruses are not alive. They need a host to be able to do any damage, and if antibodies prevent viruses from docking, then the virus can't do anything. So now let's talk about the third line of defense. This is when we actually get into specific immunity. So this is when we start talking about antibodies being present and antigens causing this reaction. Okay, so we have a number of cells that are involved with this. We have B cells, which can help with when you encounter an antigen, and they also help with creating memory cells for future infections. So the lymphocytes are involved with making these antibodies. So plasma cells are specific types of lymphocytes that make antibodies, which are a type of B cell. I know, there's a lot of cells here. And these white blood cells, these lymphocytes, can recognize and respond specifically to different microorganisms and also create memory cells. Those memory cells are useful because they don't take up a lot of space because you only keep a few in relation to how many are created in the initial infection. So they stay around in your body for years, right, after. And if you get a infection again with the same antigen, then the response time will be way less, and it'll be a really effective and efficient response because those memory cells will immediately start making antibodies again to fight off this second infection. So let's talk about the actual steps to the immune response. So you have an antigen entering the body, and that antigen is identified. A specific plasma cell, the B cell, that can actually produce this antibody is identified. The plasma cells will then reproduce or clone themselves and produce specific antibodies. Those antibodies will circulate around the body, find the antigen, attach to the antigen, and then eliminate it. And then some of those cells become memory cells, and remain behind to provide immunity, and the memory cells will then respond quickly if there's a second infection. Now we're going to talk specifically about HIV and AIDS. This is one of the examples that IB is using and will show up when asked questions about the immune response and the effect that this specific disease has on the immune system. So this is a syndrome that's caused by a virus. A syndrome is just a group of symptoms that are found together, right? So this is considered a syndrome. So AIDS symptoms are low numbers of T helper cells, which help with the B cell production, weight loss, and a variety of diseases caused by various pathogens or malignancies. So when you have a low number of T helper cells, you have a weakened immune system. And when you have a weakened immune system, fewer antibodies can be produced. So when the AIDS virus gets into, a human's immune system, it really lowers the ability for the immune system to work properly, which then leads to an easier infection rate of more common things. So if you have a weakened immune system and you're not producing antibodies, the flu is going to hit you way harder than someone who does not have a weakened immune system, right? You cannot fight off things as well when your immune system is weak. And that's what this virus does. So when we look at this, when we have this initial infection, you can see that you're totally getting, losing the lymphocyte count, right? Your cells that are necessary in your immune system, you're really lowering them out. And then they just kind of continue to lower. And so again, once T cells drop below a critical level, the immune system is ineffective, and the patient now is considered someone who has AIDS. And patients with AIDS are super susceptible to opportunistic infections, right? So things that wouldn't normally hurt them as badly, they're now super susceptible to, which is what I was just talking about previously. So when your lymphocyte count goes down, the copies of the HIV RNA is going up at the same time. And you can see this cross where all of a sudden the HIV virus is super prevalent in the body. And as time goes on, right, it kind of comes back down, your immune system might come back up. During this time when it's not really it's sort of on the rise, but it's pretty consistent, really, and your T cells are still there, but they're starting to go down. This is called clinical latency. So this is when, yes, you have AIDS, but you're okay for right now. And then as your T cells drop below this critical level, that's when the concern is even more than it was before, and that's when opportunistic diseases can really come in and do a lot of damage. And once those opportunistic diseases come in, it's much harder to get them out. And especially years ago, before there was a ton of research on this, like the 80s-ish, 70s, 80s, this would almost always result in death. Of course, now there's a lot more research, so there has been a lot of improvement on the life expectancy of those that contract AIDS, but back in the 80s when people were extremely scared about this and didn't understand it and were acting in not a great way. If you want to look more about this, you can look up Reagan administration with AIDS, and you can see how influential it was when Princess Diana actually shook someone's hand that had AIDS. It was world news because people thought that you could get AIDS from touch, and the picture of her shaking someone's hand without a glove on or anything was whoa. It was a huge deal. So anyways, we've come a long way since then, but those are some pretty interesting, and that's our past. So it's cool that we've been able to use science to improve as much as we have. Continuing on though, transmission is not through touch, not like skin to skin contact, but it is through transfer of body fluids. So if you have a small cut or tear in the vagina or penis, mouth, intestine during sex, that's when a lot of the transfers happen, which is how most people got infected with AIDS. Again, in like the 70s, 80s, early 90s, that was like the main way HIV was transmitted. You can also get it from hypodermic needles, reusing needles. It can also be passed across placenta from mother to baby, cuts during childbirth or during breastfeeding, and then with blood transfusions. So if it's in your blood, and you donate blood, and then that blood goes into someone else, well, you just, of course, put that virus directly in someone else's body. The impacts of this were very large. It spanned pretty much every aspect of someone's life. So there were social impacts and there were also economic impacts. The social impacts, this is especially true in the 80s. The discrimination and the stigma was unreal. I know you guys weren't alive then, but I do urge you to just like look up that, like what was going on in the 80s, how were people treated that had HIV or AIDS. There was a lot of abuse and harassment. They weren't able to find work because once an employer found out about this, they would typically just like remove you from the workforce, or they weren't healthy enough to work, which then of course impacts economically, but also socially, because now like you don't have a job, which is where like adults spend a lot of their time. There's no money coming in, so you can't like go out at night because you don't have any money to spend. The expense of medication alone could like bankrupt families. Of course, there's the risk of spreading, and then the burden of care for those that are taking care of the sick. There wasn't a lot known about it, and as mentioned earlier, this typically ended in death, and so there was a lot of, you know, this burden on people taking care of their loved ones that had this, that impacted them socially. The economic impact, of course, was the working age population is reduced. If they're not able to work or they're not finding work, then there's not people working, of course, which means that the population that, you know, runs the entire economy has been reduced. More children have to work because of that, right? If, like, the people that are, like, 25 and older are not working, then the younger people have to step up and start working, so less of them are in school, so that means the education of them, of those students that are, or those younger people that are stepping up to go into the workforce, they're going to be lower, right? The education and the graduation rates are going to be lower, so the education across the nation suffered. Of course, heavy economic burden of care with medications and medical expenses and all of that, and then an international stigma impacts trade and industry. So if there's like, when this was less understood, if there was a huge population in a certain country that, you know, it was like HIV and AIDS is super prevalent in this country, then nobody wants to trade because they don't want to have the possibility of transferring this virus along with any of the things that they're buying from that country. So that impact would, you know, that's a countries make most of their money on trade and industry. So if you're not trading with anyone, then you're not making any money for the country. We're going to talk about antibiotics now. So antibiotics kill or inhibit the growth of bacteria by targeting specifically the metabolic pathways of the bacteria. So they can target key enzymes, the ribosomes, the cell wall, whatever they need to target to prevent the bacteria from continuing on. So there are two types, right? They can be bactericidal or bacteriostatic. So when an infection occurs, the bacteria is going to reproduce, right? Of course, it wants to take over the body. If you have a bactericidal type of antibiotic, what it does is it just comes in and kills the bacteria. And it's like, nope, not dealing with this. Like, goodbye, get out of my body, please. And so that bactericide is just going to come in and like basically like nuke the bacteria, just like totally kill them. And then the macrophages come in and clean up the infection, but the bacteria like pretty severely drops, right? So it's a shorter time frame. If you get the bacteriostatic type of antibiotic, when you get the infection, bacteria grows. The bacteriostatic just means that it prevents the growth of the bacteria, so it kind of keeps them in this like latent phase where they exist, but they're not growing in numbers, and as they're just existing, it takes longer, but then the lymphocytes can come in and kill the pathogens, and then again there's a cleanup. So this takes longer, of course, but you're kind of like still having the same initial amount of bacteria. It's just whether the antibiotic is actually killing them or it's just preventing the growth, and your immune system itself is killing the remaining bacteria. So antibiotics and viruses, well, viruses aren't alive, they don't have the same structures, and they don't have any of the structures that are targeted by antibiotics. So antibiotics, bio means living, antibiotics do not affect viruses. There are antivirals, which are similar to antibiotics, but they're obviously effective against viruses because they target different things. So they target specific proteins or a specific stage in the life cycle, or they can stimulate the immune system to prevent replication of the virus. So antivirals are different than antibiotics, and antibiotics don't affect viruses. Bacterial resistance is kind of scary, actually. So bacteria reproduce super quickly and show a lot of genetic variation. With so many bacteria, they just grow immune. They show resistance to antibiotics. So if you take antibiotics, you're going to kill off all the ones that are able to be killed off, but those resistant bacteria are then going to survive, and they're going to pass on those resistant genes to their offspring, and then those resistant bacteria can replicate and replicate and replicate, and those antibiotics that you took before are going to have no effect on them. So one way to avoid this is complete the course of antibiotics that are prescribed to you by a doctor. There's a reason why you have to, you get 10 days worth of antibiotics. There's a reason why you're supposed to take 10 days, and it's to prevent this resistance. If you start feeling better on day 6 and you just stop taking the antibiotics, this is how mutations happen and those bacteria can become resistant, okay? You can also avoid this by maintaining high standards of hygiene in hospitals and farmers not using antibiotic feeds so that you're not getting these antibiotics in like every aspect of your life and only when you actually have an infection so that it kills it right away and it doesn't lead to any resistance. For our last bit, we're going to talk about Florian chain. Florian chain kind of built off of Alexander Fleming's experiment. So Alexander Fleming discovered penicillin in 1928. Penicillin is a very, very common antibiotic. Florian chain developed a method to grow penicillin, which is a fungus, in liquid and then isolate that medicine. So they tested penicillin on 8 mice. They gave pneumonia to the mice, and then in 4 mice they gave the injection of penicillin, and 4 mice they didn't give the injection of penicillin, and then the mice died that didn't get the injection. Again, keep in mind that this is, the ethics were not there in the past, and you can see that very clearly based on old experiments, but this is how we first tested penicillin, and then we tested it on humans. A policeman with a scratch on his face improved when they put penicillin on him, but then they ran out of medicine and he died. So again, not a great start. And then they tested, but it did show that there was success with using penicillin on humans, and so the next bit they tested 5 different patients that had infections. All of them were cured, but one did die from a brain hemorrhage related to the infection, but that wouldn't have been cured by penicillin anyways. So once it was kind of shown that, yes, there is very clearly a relationship between penicillin and the ability to cure infections, pharmacological companies began to produce it, and more extensive testing was done. And now it's like the most common antibiotic that's out there. Okay, so great job. I know this was a little bit long, but there's a lot to learn about defense, and so I can't wait to get started on this in class.